Okay, so I think we will crack on, seeing as we've got a good number of people here. Hello, everybody. For those of you who don't know me, I'm Pauline McGuigan, and I'm one of the members of the camera team from the Department for Health. And it is my very great pleasure today to introduce an old colleague and friend, Dr. Emma Hodson Tole from Manchester Metropolitan University. Emma's a reader in neuromuscular integration. Her work has explored how the neuromuscular system functions from the very smallest motor unit within the system up to the whole muscle group level. And she's done this in a range of different animals, from rats and horses during her PhD at the Royal Veterinary College to cats during a postdoc position at Georgia Tech, and then finally onto humans when she started her Sir Henry Wellcome Fellowship at Manchester Metropolitan University a few years ago. She's now progressed up to reader there, and her work, her recent work, is really sort of pushing the frontiers of our understanding of how muscle and the neural input to muscle makes us function. She uses a combination of ultrasound and very clever electromyographical techniques to understand how muscle works, and this work is being applied in the diagnosis and investigation of neurodegenerative diseases and other diseases of the musculoskeletal system. So over to you, Emma. I'm very much looking forward to this, and I'll be back at the end to chair the questions.
Okay, well, thank you firstly for that great introduction. I'm very flattered. And yes, it's always interesting when you hear people who've known you for a while kind of sum up what you've been involved with. So can you see my screen okay?
Yeah.
Yeah, one thing I am just going to do is I do have some videos that I hope are going to play nicely within this. So what I am going to do is I'm just going to switch off my camera so that hopefully the bandwidth doesn't get too overloaded, but I will come back into view towards the end, obviously, for questions. Okay, so as Polly's really nicely summed up for me, my interest in terms of my research background is really related to how we move. And it's really clear to us that physical activity is such an important determinant of our quality of life, no matter what age we are. And in order for us to complete any physical movements, be that moving through our physical environment, be that speaking, swallowing, breathing, they all require appropriate functioning of both the nervous system and that to be integrated with the skeletal muscles. So much of my work has focused on developing tools whereby we can better study those behaviours and those properties of the skeletal muscles and their connection to that nervous system, in part because there's so much that we don't understand about skeletal muscle function. So the two kind of main modalities that I've been interested in using and developing, firstly is electromyography. So this is just a sort of a general example signal here. And electromyography, or EMG, gives us the ability to record the excitation of that muscle tissue. So it gives us a sense of the neural drive to those skeletal muscles. And that neural drive results in those muscles changing shape. So they'll contract, change shape, and the forces generated at the level of the fibres get transmitted through the muscle tissue, through the tendon, and to the joints, and the joint torques that we need to move the skeleton and move through our environment. And we can capture some of those muscle shape changes using ultrasound imaging. So I've got an example here of an ultrasound image that I hope is playing for you. So this has been recorded from the medial gastrocnemius muscle. Oh, 1 second. Sorry, that was the necessary academic dog intervention. But we have here an ultrasound image recording from the medial gastrocnemius muscle. So this is located in the calf region of the lower leg. And we can see on the image here, the skin surface is along this boundary here. The muscle itself is situated in the central portion here, and then we've got some deeper muscles shown here as well. And how the muscle moves and changes shape has been the focus of quite a lot of work, because it relates to that force production, that force translation, if you like. So in the kind of biomechanics world, we've been really interested in capturing the length and the shape change of the fibres or the muscle fascicles that reside in the muscle belly. So in this region here. And the main reason I moved to Manchester Metropolitan was really to gain expertise in this imaging approach. And the work we've been doing has really looked at developing new ways in which we can computationally analyse these images and extract as much information as possible. So in the example you can see here, just to focus on this, we've got the yellow lines on the outside of the image here and here have been identified using an active shape model based approach, and the blue line through the centre here is representing the mean fascicle length and orientation through the whole of the image, which we've identified using a Bayesian hypothesis based approach. And this gives us some of the tools that can help us explore muscle mechanics, muscle behaviours during different dynamic tasks. So, for example, we can have a look at just postural balance control. So again, this is the same muscle being recorded, but you'll see the movements here are much, much smaller. And these are occurring just as the person is stood quiet standing balance, and you can see we've got some subtle changes in both the fascicle lengths shown on this top graph here, so this blue line through the muscle belly, and in terms of the muscle thickness, so the distance between the top layer of the muscle and the deeper layer of the muscle. So posture, it has these quite kind of small changes, but when we think about more dynamic tasks like running, we can see much larger changes. So these are images that were recorded again from muscles in this calf posterior compartment region of the lower limb during running. And you can see, again, the active shape model based approach has been used to segment these images, identifying the deeper and superficial boundaries of one muscle here, the soleus, and a muscle closer to the skin surface, the lateral gastrocnemius. And we can explore these measures of thickness and understand how the muscles might interact in terms of sharing their space. So how much do they bulge? And this was a piece of work that I recently completed in collaboration with Dr. Adrian Lay. And we explored how those thickness changes occurred in these muscles sharing this compartment in the posterior part of the leg, and how where in the image we measure those thickness changes might influence our interpretation of what the muscle is doing. Now, I wanted to kind of start off sharing these examples with you, just to give you the kind of background, the idea that we have tools that enable us to quantify the kind of traditional measures of dynamic skeletal muscle behaviour related to measures like thickness and the orientation and the lengths of the fascicles. And we can do this more or less non-invasively using these ultrasound imaging approaches. But as I said, these measures are quite traditional in terms of what we've been interested in from a mechanical perspective. And as we've worked more and more with these type of data, we've started to become aware of the fact that actually there may be many other useful information contained within these sequences. One of these that I'm going to focus on now for you is really thinking about how the nervous system connects to the muscle tissue to create those movement dynamics. So we've already talked about this connection. So our voluntary muscle movements are related to activation of the nervous system. And at the periphery in the spinal cord, we have alpha motor neurons. So these are the cells that connect to the central nervous system. Their axon will lead down to a muscle, and each motor neuron will innervate a family, a group of muscle fibers. And when a motor neuron fires, those fibers will contract together. They'll contract in tandem, synchronously as a group. And we term this the motor unit. So the motor unit is the alpha motor neuron, its axon, and the group of muscle fibers that are innervated. So those fibers will always contract together, receiving the same command signal from the parent motor neuron. Now, when motor units receive these commands, obviously in the cartoon you can see I've simulated some kind of movement, and in the ultrasound images that we can record, we can also see localized displacement within the tissues that we image. So hopefully this is coming through, that you can see again the main muscle I'll focus on is the medial gastrocnemius again here, and in the image sequence hopefully you can see a yellow box will come into view, and then in that yellow box you'll see a sort of a transient movement of the muscle tissue. And what we're seeing here is involuntary activation of motor units by the nervous system. Now, this happens quite frequently in healthy individuals. It's associated with fatigue. It's associated sometimes with overstimulation. So there's an idea that we see more fasciculations if people have a high caffeine intake. But hopefully, through this view, you can see that they're occurring fairly randomly. They're not occurring with a kind of very distinct pattern, which is quite different from the kind of regular pulsation that you can see coming through in this left-hand border of the image. So this movement that you can see happening regularly here will relate to blood vessel pulsations. And then on top of that, we have these localized displacements occurring related to excitation of these individual motor units. Now, we've become really interested in this for a couple of reasons. One, related to kind of fundamental questions about motor unit properties, but secondly, because of a potential clinical application which relates to diagnosis and monitoring of a particular neurodegenerative disease called Motor Neurone Disease. It's also known as Amyotrophic lateral sclerosis, or ALS. Now, motor neurone diseases, or MND, are a fatal, rapidly progressing family of neurodegenerative diseases that affect the neuromotor system. They are incredibly rapid in the sense that within approximately 18 months of diagnosis, about 50% of patients will have died. And part of the progress of this disease really highlights to us the importance of appropriate neuromuscular skeletal functioning. And this is because people living with MND will suffer from a range of symptoms, from slurred speech to difficulty swallowing, shortness of breath, difficulty grasping objects, as well as difficulty standing and also walking. So typically there'll be a focal onset. So it might be that someone has a fall and notices they're not as able to walk as they remember. They may have, or another individual may have difficulty picking up an object. Others will notice that their speech will become slurred as they become tired. But on the whole, patients will see all of these symptoms occurring. And as the disease progresses, most patients will pass away related to the changes in the respiratory system and those respiratory muscles. At the moment, there's no effective treatments. There's one licensed medication that has been shown to extend life by approximately 3 months. And therefore, there's a real need for sensitive biomarkers, particularly to support effective clinical trials where maybe drugs that have been approved for other diseases could be reappropriated. But without sensitive markers, the scale of trials that are needed are quite prohibitive in this relatively rare, but obviously rapidly progressing disease. So one of the signs of this widespread denervation is something called fasciculation. And these fasciculations are exactly what I showed you on the previous video. They're involuntary activity of individual motor units. So as part of the diagnosis process for MND, they're looking for these indicators of ongoing denervation, and fasciculation is seen as an important component of this. So it does form part of the diagnostic criteria. Now, currently in the clinic, those fasciculations are identified using needle-based electromyography. Now, because the clinicians are looking for these systemic changes, essentially that means that patients will have to undergo needle-based exploration of muscles across the whole of their body. So they'll investigate all the main sort of nerve branches through the legs, through the arms, through the torso, the thorax, and around the head and the neck region, including the tongue. So it's a really extensive investigation. And firstly, many people don't like needles. And secondly, needle-based EMG may not be the most sensitive way of identifying these fasciculation events. So there's been a huge amount of interest in identifying whether ultrasound imaging may be a useful alternative or complement to some of the data that we can extract from electromyography. So you've already seen, and there's evidence out there in the general literature that fasciculations are visible in these ultrasound image sequences, and we've known that since the 1990s when Walker et al. Kind of first identified this. More recently, it's been shown that ultrasound can be a more sensitive detector of fasciculations than EMG in some muscles, which I think also includes the tongue. But there's a but, and the but is all of the work to date has been based on manual observation of these ultrasound image sequences, and therefore relies on clinicians or on observers identifying the presence or absence of fasciculations in the image sequences. And that's problematic for a number of reasons. Firstly, when we think about consistency in terms of consistency between observers, between different sites conducting studies, and it also poses a problem for keeping detailed records over long periods of time. And from I guess a more sort of scientific perspective, it also means that we're not taking the most, we're not extracting or exploiting the most amount of information that's available within these ultrasound image sequences. So over the last few years, we've been looking at trying to change that situation. So how can we go about doing that? Well, we've got kind of a framework that we've been working within that has three main pillars. We can think about the information sources. So where can we understand what's happening in those ultrasound data, or what's happening in the muscle, I should probably say first of all. And those information sources include the ultrasound images themselves. So obviously we can manually observe them and see what's happening. And we can also use those clinical tools. So the neurophysiologists will use intramuscular electromyography, but there are also means of recording surface-based signals, both using single and multi-channel based technology, which I'll talk a little bit more about later. But these can provide us information about what's happening in the skeletal muscle, the behaviour of the muscle. We then need to think about what computational approaches we want to identify and develop. So what algorithms might be appropriate? And obviously we can use our information sources to help inform that algorithm identification, and then we can go forward and implement them, and therefore need to evaluate their performance. So how well are they performing? And for that, we obviously need some kind of ground truth. And this again is where our information sources can come in. And then over iterative periods of work, we hope we can come up with some kind of best practice application. And that in itself can also move us forward in terms of providing us with a computational analysis that gives us a new information source that we can feed into the process. So for our first steps, we focused on using manual observation of the ultrasound images. And we used a feature-based tracking approach. So I'm just going to talk you through that first study. We recruited 20 healthy adult volunteers, and we also had 5 patients who had previously been diagnosed and were living with motor neurone disease. We collected data from the biceps brachii, so in the forearm, and the medial gastrocnemius in the lower leg. And you've got a couple of example images here from unaffected individuals and a person living with MND. So essentially the people just have to stay relaxed, sit in the chair whilst we make the recordings that we need. And from those recordings, we did 2 analysis steps. Firstly, we had 2 observers independently identify the occurrence of these fasciculations, these muscle twitches in the ultrasound data. We also completed a computational analysis approach combining feature tracking with a mutual information analysis. So for the feature tracking, we placed a 100 by 80 grid of Lucas Canade features onto the image and tracked the position of those features into the next frame. So this provided us with a 2-dimensional vector grid representing the magnitude and direction of feature movement between consecutive frames. We then used mutual information to convert the 2-dimensional grid of movement vectors into a 1D signal. Mutual information is a statistical measure of the interdependence of 2 signals based on joint and individual probability distributions. So what we were assuming here was that when a twitch event happened, we would see large coherent movements of features between consecutive frames. And then the rest of the time when the muscle was quiet, that coherence would drop. So we expect to see spikes in the mutual information when a twitch occurs or a fasciculation event occurs, and the rest of the time the signal to be reasonably flat. So this is an example from that data. We've got time along the x-axis and the mutual information metric on the y. The mutual information signal is in blue, and overlaid you can see the red marks represent the identification of an event by one of the operators. And you can see on the whole we've got some really strong agreement between the 2 measures. So if we have a look more broadly at the wider data set, we really see quite good agreement. So these data here are receiver operator characteristic curves. On the X, we've got 1 minus specificity, and on the Y, we've got sensitivity. The graph on the left here is from healthy individuals, and the one on the right represents the MND data. And these two data are from the biceps brachii muscle. So for ROC curves, what we're looking for is for the line to essentially track up the y-axis and then across the x. So we essentially get a very high area under the curve. And those area under the curve values are shown here in the brackets for different combinations of signals. So we've got operator A and B, and then the union and the intersection of the two operators. Agreeing with the mutual information-based metric. And you can see for both the biceps, and we've got the same data here for the medial gastrocnemius, that we had really high levels of agreement, both for the individual operators and also their union and intersections of their data. But you can also see that we have some discrepancy between these data sets. So we were really happy with these results because it was really the first time that we were able to show that a computational approach could be used to automatically identify the occurrence of these fasciculation events in the ultrasound data. But one thing that we did note was the fact that we've got much higher agreement between the manual IDs and the mutual information in our healthy data. So if we look at these two values here compared to the data from the people living with MND. So this did lead us to kind of think a little bit about, well, why might that be the case? What's happening here? And a few ideas have come to light. So firstly, for this work, as I said, we used Lucas Canade features in a feature tracking based approach, and that depends on there being features available to track in the images. And we know it's well documented. So Maurits back in 2003 showed that the intensity content of the image sequences differs between different pathologies, so different neuropathies and myopathies. And you can see here in our healthy individual, we've got really clear distinction. Fascicle lines in the muscle. This is the same muscle in the same region in a person living with MND, and you can see that that distinction is really, really lost. So the changes in the muscle tissue that are occurring as part of the disease process is influencing maybe some of the properties of the image that will have a detrimental effect on the feature tracking based approach. And that will also be influenced by the orientation of the probe, the ultrasound transducer, relative to the muscle. So whether we line the muscle, the probe along the long length of the muscle, will give us different images to when we place it cross-sectionally, so transversely across the muscle. So the image content will be influenced by this transducer positioning as well, and that can have an impact in terms of how well tracking based approach might work for different operators collecting the data that is used. Another element that we identified based on using the mutual information based approach was that we were picking up other physiological events in the ultrasound data. So here we can see really clear spiking in the mutual metric. But these spikes are obviously really, really regular, occurring with regular intervals. And here, rather than capturing fasciculations, we've got an excellent monitor for blood vessel pulsation. But obviously we don't want this when we're trying to identify the occurrence of fasciculations, and this could impact the accuracy of any interpretation. Equally, when we start to have a look at other muscles away from the limbs, so those closer to the thoracic region, so those potentially more influenced by breathing patterns, we can start to find patterns where the mutual information is not able to identify the coherent spikes outside the kind of background of breathing patterns that are occurring. So whilst this was a really exciting start for us, and we feel like we've made really good progress, particularly in terms of making it clear that computational approaches could be valuable within this kind of clinical question, there's obviously still more work to be done. So in the next kind of iteration of this project, what we wanted to do was move away from the manual observations and start making use of measures that were more clinically appropriate. And we've specifically focused on using intramuscular EMG, as that is the clinical gold standard at the moment. We were also aware that we wanted to move away from feature tracking based approaches and start to explore whether there were other measures, other approaches to analysing these image sequences that might be more robust, particularly in the face of the changes that are occurring due to the pathology, as well as the potential influence of the probe positioning as well. Now, it was at this point that we were fortunate to secure funding from the MND Association, which is the UK's only charity that focuses on MND research at that time. And we were able to recruit Kate Bibbings, who came to do her PhD training with us, funded by the MND Association. And so this kind of second step really lay to Kate to move forward for us. And she's come up with some really exciting ideas, which I think have really moved us forward. So her suggestion for moving away from the feature tracking based approach was to use more of a learning based approach called foreground detection. So within this method of working, what we're assuming is that the fasciculation events are causing transient changes in the pixel intensity. So again, if we go back to this video that I was showing you earlier, if you look at what's happening within those boxes, those yellow boxes as they come into view, we're identifying a movement because the pixel intensity values in that region are changing. And those changes we can think about as being foreground events. So they're happening in an isolated area and are different to the rest of the image, which is maintaining its characteristics. So we can think about the rest of the image being background and these fasciculation events, these disturbances in the pixel intensities, as being foreground. So for foreground detection, we need to be able to classify our pixels as either being part of that foreground event or as being part of the background. And we use that using Gaussian mixture models. So for each image sequence, we'll use the first 500 frames as training data to develop or define a mixture of Gaussians that explain the pixel intensities that are seen. So in the diagram here, I've got an example from one pixel. The grey bars in the background give the frequency distribution of the pixel intensities that are seen. And this frequency distribution is explained by 3 Gaussian models. So we've got the red is distribution 1, the blue is distribution 2, and then out on the side here, we've got a third distribution in yellow. So because the background is what we will typically see most of the time, and the fasciculations are much, much rarer events, we can classify pixels whose when this pixel intensity falls within one of the first 2 distributions, we can say that that pixel is part of the background. We can classify it as a background pixel. However, when its intensity falls into the 3rd distribution, we can say that it was part of a foreground event, which we will interpret as being a fasciculation. Now, the advantage of this process is that these models, these mixture models, are updated over the course of the remaining images that are fed into the process. So whilst we establish the model on the first 500, it continues to learn on subsequent frames, which means that events like the blood vessel pulsations should be learned to be part of the background, hopefully keeping these lower weighted distributions as just identifying foreground and those relating to fasciculations. So to test this approach, we collected data from 19 healthy participants and 20 people who had been diagnosed and were living with motor neurone disease. We collected data from 5 different muscles. So I expanded away from the legs and the arms. So we had biceps brachii in the arm, medial gastrocnemius in the leg, as well as 3 muscles across the trunk, the back. So we've got rectus abdominis in the thoracic region, the paraspinal muscle at the level of T5. So this is actually on the back. And then the trapezius muscle, the cervical portion of the trapezius muscle. And we were simultaneously recorded intramuscular EMG using the needle-based approach and recorded the ultrasound images as well. And these are the results. So Kate tested her Gaussian mixture model foreground detection approach alongside our previous feature tracking mutual information based data or approach. And again, we're using receiver operator characteristic curves to explore the agreement between the computational output and this time the intramuscular EMG detection. So I've got 2 columns of data here. So the biceps brachii data are shown in this column, and in the second column on the right is the gastrocnemius medialis data. Data from our healthy participants is in the top row, and then those who are living with MND is in the bottom row. The blue relates to the mixture models, the foreground detection, and the red relates to the mutual information based approach. And then the solid line relates to transverse view. So we had the probe orientated across the muscle, and the dotted line relates to where that transducer, that probe, was aligned along the length of the muscle. And you can see in all of these, again, we've got the area under the curve this time as a percentage is shown on each of the graphs. And hopefully you can clearly see that the foreground detection approach has outperformed the mutual information based approach in both muscles and also in our healthy and MND affected individuals. The other 3 muscles, we weren't able to look at healthy individuals because fasciculations didn't occur in these muscles in our healthy people, which was, I guess, part of also a confirmation that they were definitely free from the motor neurone disease. So these are just, and we also couldn't complete the mutual information based approach because each of these muscles, so the rectus abdominis shown here, the thoracic paraspinal shown here, and trapezius shown here, they were all affected by breathing patterns, which meant that we just weren't able to detect fasciculation related spikes in the mutual information. But what you can see is that the accuracy of our foreground detection has been quite high in each of these muscles as well. So overall, we were able to see an 80 to 92% accuracy for the foreground detection approach versus this clinical gold standard of the intramuscular EMG. And importantly, we know that this objective measure works in each of the body segments needed for MND and ALS diagnosis. So we really feel like we've taken quite a significant stride forward here in terms of being able to develop a tool that is going to have clinical use for diagnosis and potentially for monitoring disease progression as well. But again, you kind of always end up at the end of a project, or at the end of a phase of a project, I should say, coming up with other questions. And we kind of go back to this idea of, are we really extracting all of the information that might be available in these image sequences? And that brings us to the idea of, well, what other things about the fasciculation might we be able to quantify? Now, the foreground detection gives us an opportunity to maybe look at something new. So in the sequence that should be playing now, you'll see there'll be transient yellow points coming in onto the image sequence. And these represent the location of foreground objects that the foreground detector has identified related to a twitch. So underlying that, you'll see the muscle tissue is moving. And these points represent where foreground detection has identified the pixels are associated with this fasciculation event. So we can start to look at some of the characteristics of these foreground objects. How has the tissue moved? How has it been displaced with the fasciculation event? And Kate went on to explore some of these spatial properties. So essentially using connected components to measure the longitudinal distances. So essentially along the y-axis, what were the distances across those foreground pixels, as well as along the x-axis, what were their distances. And also quantifying something called the Euler number, which essentially is a measure of the number of holes in the object. So that kind of gives us an idea about how spatially clustered or more widely distributed these foreground pixels were. And if we plot these three kind of measures, we can come up with some really interesting data. So these are the data that Kate had collected. We've just got the two muscles here, so the biceps brachii on the top and the medial gastrocnemius on the bottom. Two muscle views, so on the left here are when the probe has been placed transverse, so cross-sectional view through the muscle, and on the right here we've got the longitudinal view, so the probe's placed along the length of these muscles. The red points relate to MND affected data, and the blue points are data from our healthy controls. And you can see in some of the muscles and muscle views, there's no distinction between the distribution of these kind of spatial measures. So I should say we've got the total X and Y distances, so those longitudinal and transverse distances on the axes coming across here and on the vertical, and then the spread along this axis relates to that Euler number. And when we look at medial gastrocnemius in the transverse view, we actually see no separation between the behaviours or the spatial properties of these twitches between our patients and the healthy controls. But when we look at some other muscles and other views, so for example here the biceps, we can see really clear clustering, really clear differences between our MND affected data points and those from the healthy controls. And in the medial gastrocnemius, when we view the longitudinal view, again, really distinct clusters between the two groups that we've studied. So this suggests that the tissue displacements that are occurring between healthy and this neurodegenerative disease have different spatial properties that it may well be useful for us to start exploring quantifying. This is because it may relate to features of denervation and reinnervation that happens as part of the neurodegenerative process. Equally, it may relate to differences in terms of how the tissue is actually composed. So as part of the degenerative process, we'll start to see fat infiltration and fibrosis in the muscle, which could affect how these the forces at the fibre level are translated through the muscle tissue to provide the kind of displacement patterns that we are seeing. And this also has potential application for studying in new ways how the muscle changes as a factor of other processes, of which aging would be something that's really of interest, because we know motor units change, muscle properties change, and this has an impact on force producing and force control capabilities. So what are the next steps? Well, one question we have is whether intramuscular EMG is actually the most useful thing to look at. So if you look at the video here, the red signal is in the intramuscular EMG, and you can see through this period there's really nothing detected at all. But you can very clearly see in the ultrasound data and in our measures of fasciculation events that fasciculations are occurring. But our intramuscular EMG has been too sensitive, it's too localized a detection volume to be able to identify the occurrence of these twitches happening. So one thing we're really interested in exploring is using high density surface EMG arrays to detect the fasciculation events, and coupling these with the ultrasound imaging so we can more tightly bring together the excitation, which is happening through the grid now, and the resulting tissue displacement that occurs. And together with the group in Torino, the work I've been involved with has mainly been with Dr. Alberto Botta. We have developed a grid of electrodes that we can place on the skin surface that's transparent to the ultrasound, so we can lay the transducer directly over top of these electrodes and therefore simultaneously record the activity, the electrical activity and the resulting tissue displacement that's occurring. And I've got some work with clinicians based at King's as well as at UCL exploring how we might be able to couple these things to understand the electrical and mechanical coupling within the muscle of our patients, as well as trying to track longitudinal changes, so how the disease progresses over time. So just to summarise for you then, hopefully I've been able to build a picture that the ultrasound image sequences are a rich source of information. And within that, moving away from kind of traditional mechanical measures, we can we can extract motor unit behaviour, which has use in terms of detection and monitoring of neurodegenerative disease progression and potential application for understanding some of the changes with ageing. And I'm really excited about maybe moving forward to help us better understand the kind of relationships between how the muscle is excited by the nervous system and how that resulting activation translates into torque production. And I think we've got a real opportunity to explore that with these sorts of tools. So very finally, obviously this work doesn't happen alone, so I just need to acknowledge my collaborators. You've already hopefully seen Kate, but Dr. Nick Coombs is the clinician who's supported us on the project. Ian Loram, Pete Harding and Nick Costin were heavily involved with a lot of the work, and obviously we've had funding from a range of sources that have helped us at various stages along the way. So at that point, I shall stop sharing my screen and switch my video back on, and happy to answer any questions that anyone might have.
Brilliant. Thank you so much, Emma. The thing I really missed here was applause. Applause via video call feels a bit odd, but I'm sure if we were all in a room together, there would be a rousing round of applause at this point. Thank you very much. Beautiful graphics, beautiful images, and really clear explanations. Thank you. Do we have any questions from the room, as it were, the virtual room?
This is where I realise that maybe there are other people out there. I've just been chatting away to myself in the room.
Any questions? I've got one to crack. I've got one to crack us off.
Go on then.
In terms of, so I think that the images are beautiful. It's so clear to see kind of what's happening that makes it really, really tangible that you're then doing the computation analysis of that. I mean, what is the sort of time resolution of these events, and therefore what kind of technology, ultrasound technology, do you need in order to be able to detect them?
Sure. So the twitch events, they happen over around about 500 milliseconds. So they're much, much longer than the excitation events. So with EMG, we're typically, well, for intramuscular, we're typically sampling at about 5,000 hertz. But obviously and we're trying to capture events that are kind of 25 milliseconds long. But the resulting tissue response is much, much longer. And we're really looking at something more around sort of 500 milliseconds. So we're able to capture that with our standard focus B mode based systems. The one that we've relied on is the telemed system. So we've been capturing at around about 80 frames per second for most of our data. Now, that doesn't mean to say that there's probably not additional information you'd be able to extract with a higher frame rate. But most of this work has been done with an eye to what the clinicians might be able to have access to, and therefore those kind of standard B modes are widely available throughout the healthcare system, which has been one of our kind of driving focuses in terms of trying to develop tools that might easily translate into what's already out there, if you like, or to equipment that's already widely available.
And is this a tool that's going to be able to produce an immediate result for a clinician?
So the mixture models, the foreground detection is pretty rapid. It doesn't take that long to complete. Now, I'm not saying that we're at the end of this journey, because there are some issues. So one element is that the foreground detection, it does need some parameterization. And we've been exploring, since Kate was with us, I've had a number of other data sets that have kind of helped me explore more of what those parameters might be. So I think what I probably should have said in that summary is that we're not at the end of this journey. I'm not kind of saying, well, this is the thing that's going to definitely get out there. I think we're still exploring, and there's still opportunity to find other approaches that will outperform, again, for different reasons maybe. You'd be close to being able to turn something around within a clinically relevant time frame with what we currently have.
Brilliant, thank you. I think Neil has turned his camera on, therefore I'm presuming he has a question.
Brilliant, thanks very much. Thank you it's fascinating. I should say from background, I'm from computer science, so I have a vague understanding of the signal processing parts of ultrasound, which I sort of looked at a bit before. I have zero understanding of anything that anyone might look at with an ultrasound machine. So take it from the machine learning point of view. But I was just wondering about the statistical models that you're fitting with the Sodic Gaussian mixture model. Are these tend to be operating per frame and things like that, or are you tracking the statistics over time and things like? Is there a temporal component to the frame?
So the models, I think, so they'll be applied. So the initial models will be developed. I don't think it will matter what order those initial 500 images go in for the training data. But obviously for the categorization into foreground and background, we want to know the time point of those events. So they're applied over time, and the learning that will happen to update those models will therefore occur over time, or with a time-based component in there.
But the model itself is a sort of, so I guess you've answered with the training data, you look at it as snapshots of time for the capturing the statistics, things like that. Because it just seems very well, I suspect other people involved in the project, I see things here, but will say more things than me, that there's interest in certainly in from a friend of mine who works on making ultrasound machines of what sort of new ultrasound machines can be used and need to be made. And I wondered if sort of things like being able to trigger higher frame rates, or being able to capture
Yeah. Well, if you're going,
like that,
if you're going down that route, so one of the challenges with the ultrasound is obviously we just have that single plane through the muscle tissue, and those the fibers, the fascicles will sometimes they'll be exactly in that plane, sometimes they'll be transitioning through the plane, and therefore when it comes to understanding the displacements that are occurring linked to that excitation, there are challenges there. I think I've probably thought about these more in terms of our fundamental questions. So thinking about, what does an EMG signal from a motor unit look like? How is that kind of spread through the muscle tissue? And what does that mean for how the muscle responds? And to do that really well, we need to start moving towards 4D, so those kind of three-dimensional views of time. But we don't have, or I'm not aware of the technology where we can have that higher time resolution as well as those three-dimensional measures. So if your friend was interested in kind of exploring those sorts of avenues, I think there'd be a lot of people that would be super, super interested in that. And it's really where the excitement of having the link between the excitation and the resulting tissue displacement. I think we're able to start probing those questions about how the two are connected, but we won't be able to pick up that fuller picture until that ultrasound technology has advanced to enable us to take those more higher temporal resolution at volume based measures, and so I've seen some really nice array based probes which are now flexible that you can mold over the muscle. So I'm waiting for that moment when I can wrap a probe around and get these sort of 4D data to complement what we're seeing in the EMG, because there's some mismatches, particularly when we start looking at some of those EMG data. We'll see things happening in the EMG that we don't necessarily see in the ultrasound, and vice versa. So really understanding what's happening there, I think it really opens the doors up for us understanding what the muscle does, because it's a black box. So it's really ripe for exploration.
Thank you very much.
Good. Thanks, Emma. There's a question from Mike in the chat, which leads on very nicely from that discussion, actually, around kind of what the power or other requirements of ultrasound imaging that make it difficult to convert that from clinical forms into something that was sort of a wearable real-time monitoring system.
Well, having said that, so we are using just sort of standard systems. They have shrunk over time, but there are now systems available based on mobile phones. So you can connect a small portable ultrasound to a phone. Now, whilst that wouldn't necessarily be wearable, I think it does make the possibility of moving some of these things out of the clinic and into a home-based environment. So particularly for patients who are weak, they're struggling with mobility issues, and they potentially don't have a lot of time either, to have something that they could do at home that could help with the monitoring, I think would be really useful. So I can't speak specifically to the exact power requirements. Ours is just a plug into the mains system, but as I said, there are definitely technological advances happening that are moving this forward in terms of making it more widely accessible.
Mike, we have within health, we have the same system that Emma is has talked about in terms of the telemed system. So if you wanted to come and have a play with that, and it's not wearable, but it is carryable at least, and kind of we've all done some stuff about kind of people moving along with ultrasound probes attached to them. So we're kind of not too far off that being possible. Any other questions? Okay, so we have gone up to our time beautifully. So it just remains me to thank Emma hugely for preparing the talk and giving it so clearly. Thank you so much. If anybody would like to follow up on any of the things, that I'm sure Emma would be delighted to carry on the conversation. But thank you very much for coming, and I hope you all have a good rest of the day and a very happy Christmas if I don't see you before then. Cheerio.
So thanks everybody. Enjoy the rest of the day.
Thanks everyone.
Steffi, could you stop the recording, please?
I can, yes.
Thank you.